Hi, everyone, and thank you for the introduction. My name is Ellen, and I'm a third-year PhD student at MIT, working in the labs of Linda Griffith and Roger Kam. And so today I'll be talking to you about my project on creating an in vitro microfluidic system for observing angiogenesis into synthetic PEG-based hydrogels from perfusible microvasculature. And I actually got switched from a poster to this talk just a couple weeks ago. And since this is the case studies session of the conference, I wanted to focus on really situating this project within our lab's larger goal of creating vascularized models of endometriosis lesions, and then talk about some of the interesting phenomena and challenges that we've encountered along the way. And so to start with some background, in my lab, we're interested in endometriosis, which is a chronic inflammatory condition defined by the ectopic growth of the endometrium outside of the uterus, where it doesn't belong, forming these structures known as lesions. And the endometrium itself is a complex multicellular tissue that's composed of several cell types, including epithelial, stromal, endothelial, and immune cells. And the growth, repair, and breakdown of the endometrium is controlled by the cyclic fluctuations of the hormones estrogen and progesterone. And so there's a regeneration of about a centimeter of tissue without any scar formation there. And what happens in endometriosis is that this endometrial growth, breakdown, and repair also happens with the ectopic lesions, resulting in debilitating pain and infertility and affecting about 1 in 10 women in the world. Unfortunately, there's not a lot that we know about the disease at the moment, and that's partly due to things like delays in diagnosis, heterogeneity in clinical populations, as well as limited preclinical animal models, and a lack of reliable therapies that go beyond just targeting symptoms of the disease. And so as a result, we think that there's a critical need for developing new techniques and model systems that allow us to really elucidate the underlying mechanisms of disease to develop better therapies for patients. And so one thing that we've been doing in the Griffith Lab over the last several years towards this goal of developing a in vitro model of endometriosis starts with creating these synthetic polyethylene glycol or PEG-based hydrogel systems. And so this is something that we've applied for a variety of different cell types in the lab, but briefly we use an 8-arm PEG macromer that is functionalized by vinyl sulfone groups. And then it's modified by a number of peptides, including peptides that or integrin binding peptides, as well as ones that sequester cell secreted ECM, as well as a peptide that cross-linking peptide that contains an MMP degradable site. And polymerization occurs under slightly basic conditions here, allowing us to create a PEG hydrogel network into which we can encapsulate cells. And so the benefit of this system that we found to be really useful with all of the different applications that we've been trying to use it for is that it is tunable and modular, in that we can really control the different cellular, physical, and molecular parameters to really tune it to what kind of cell type or tissue of interest that we're trying to culture or mimic. And specifically related to the endometrium, this is some of work that is on bioRxiv right now by Juan Necco and Alex Brown from our lab, where they've been able to create a functional co-culture of 2 of the main endometrial cell types, endometrial epithelial organoids and endometrial stromal cells inside of these droplet PEG-based hydrogels. And specifically, they identified that there are 2 major integrin binding peptides that are essential for being able to culture these cell types together. One is a collagen-derived peptide that's important for the epithelial organoids, and the other is an RGD that has a synergistic binding site that's important for the endometrial stromal cell side of the co-culture. And when we found that when you combine these 2 cell types together in this PEG-based matrix, that you can observe the function of the cells, looking at things like hormone response, stromal cell desigilization, and maintenance of epithelial cell polarity and morphology. And so this is a great step that our lab has taken toward looking at models of the physiological endometrium, as well as thinking of perturbing it for pathological purposes. But one thing that is currently missing is the concept or the role of vascularization here. And we know that vascularization is a major hallmark of lesion development due to the fact that lesions can grow up to several millimeters in diameter. And to really hit home why this is important in this disease, This is an example of a tissue sample of the endometrium is shown in this bright red stripe at the bottom, and then the myometrium, or the muscle layer of the uterus, is at the top. And what's depicted with this zoomed in portion is it's a cousin of endometriosis, adenomyosis, where the endometrial tissue ends up growing inside this muscle layer. And in our lab, we've used tissue clearing techniques through the clarity method to really visualize what this looks like. And as you can see from the red staining here is showing the vasculature, and the lesion is this it was this empty cystic structure in the middle, and it's really, we can see that vasculature is really prominent throughout the entire lesion structure. And so what we've been doing to try to really integrate this vasculature into our existing functional co-culture of the endometrial epithelial and stromal cells starts with this microfluidic device design. And so the beginning is very similar to a lot of the vasculogenesis in vitro techniques that others in Roger Kam's lab have presented on already at this conference, as well as others here. So briefly, we have a PDMS device where there are three channels, a central gel channel, and then a central gel channel, as well as two media channels that flank it. And in the central gel channel, we seed endothelial cells and supporting fibroblasts. Here, I'll be talking about using human umbilical vein endothelial cells and lung fibroblasts. And over the course of about 7 days, we can see there is network formation through spontaneous self-assembly of the endothelial cells into these perfusible vascular structures. And what's different about this system from many of the others in our lab is that we ended up keeping these 3 empty compartments, if you can see here, which have no fibrin gel and no vasculature there. And that allows us to then, at a later point in time, add our endometrial cells and surround them with this polyethylene glycol-based gel that I talked about earlier, where we've optimized its formulation for co-culture of these 2 cell types together. And the goal here is to then be able to observe angiogenic sprouting towards this lesion model that we have in this device, as well as be able to measure other kinds of vasculature-related functional metrics, such as permeability, for example. And so this is kind of the big picture of what we're working towards in our lab. But for the remainder of this talk, I'll focus primarily on some of the protocol development and platform characterization that we've done with this system, looking at angiogenic sprouting, and primarily focusing on using lung fibroblasts rather than the endometrial cells for the time being. And so the first thing we wanted to demonstrate is, can this unique geometry of our microfluidic device work, where we are able to retain this kind of empty structure in the center and have perfusible vasculature elsewhere. And so this is showing a confocal microscopy image, a bright field image on top, and then we have GFP HUVEX at the bottom, where the PEG hydrogels in the middle, surrounded by the fibrin networks. And after 7 days after adding the spheroid, which is 11 days after initially seeding the networks into the devices, we can do a perfusibility test using, here is 10 kilodalton blue dextran. And that allows us to see that the dextran can really permeate through all of the vessel structures without penetrating into that central PEG gel compartment. And so the next thing we wanted to look at is whether or not, once we add some kind of cell types into that PEG gel compartment, whether we can observe angiogenic sprouting there or any kinds of changes in the morphology of the surrounding vasculature. So here we started with using spheroid aggregates composed of lung fibroblasts and macrophages that have been polarized using IL-4 and IL-10 toward an anti-inflammatory phenotype. The rationale there was that we know that those are pro-angiogenic cell types and can be used for our initial platform development when we're exploring for the first time kind of combining these 2 types of hydrogels together in the system. And so to orient you again, this is a bright field image showing the spheroid is in PEG, and the spheroid itself is the dark mass right here. And then on the outside is the fibrin, and the networks are there. And then, so here I'm outlining where the spheroid was. And again, using GFP HUVEX, we can see that over a few days of culture, there is sprouting angiogenesis occurring right in the local vicinity of the spheroid. And then if we go further out, so this is now 11 days of culture after adding the spheroid initially, zooming into this portion, we can see there's even some anastomosis occurring some of the sprouts. And this is a time-lapse video, just really putting all this together to visualize the sprouting that's occurring. And so this right here is, these are all maximum intensity projection images. So we wanted to really confirm, is the sprouting happening going into the spheroids and into the gels, or is it just, perhaps migrating on the interface of the gel at the bottom or along the bottom of the device? So to look at that, we used a version of our PEG hydrogel that has a fluorescent peptide incorporated. So now the PEG is all of this green around the edges. The spheroid is the dark mass in the middle, zoomed in, and then the HUVECs are now RFP. And using an orthogonal view here, we can look at a slice of the XY plane and then see that there's overlap of the, the red and the green, showing that there's sprouting indeed going into the PEG hydrogel. And then similarly, when we're looking at an overlap of the dark region where the spheroid is in the green, that there is some kind of sprouting that appears to be happening into the PEG hydrogel itself as well. And then we were also curious about, since I talked about our ability to really tune the different parameters of our PEG hydrogel, whether we can alter some of its properties. So as an example here, we looked at the biophysical properties, primarily thinking about the PEG weight percentage or the polymer concentration, as well as the cross-linking ratio. So here, this is the same image that I showed 2 slides ago. Now I'm annotating that we used a 3 weight percent PEG gel with 40% crosslinker here, and overlaying the GFP in the bright field so you can see where the spheroid is. We found that if you decreased the crosslinker concentration, we got more extensive sprouting occurring in the vicinity of the spheroid. Whereas if we increased the PEG weight percentage, creating a stiffer gel that is now with an elastic modulus on the order of 2 kilopascals rather than 300 pascals, there is significantly less sprouting occurring just right there in the vicinity of the spheroid. And so some other things we wanted to try to explore with the system, and this is all some more recent data, is that we were interested in seeing what exactly is the role that maybe the macrophages that we initially incorporated into these spheroids is playing versus if we just used fibroblast spheroids itself, since there's definitely been a lot of foundational work demonstrating the pro-angiogenic nature of lung fibroblasts themselves. And so here along the top, I'm going to be showing the original fibroblast and macrophage spheroid combination that I talked about earlier, while along the bottom, it will be fibroblast only spheroids. And now here the spheroids are labeled in red. And we basically saw over the course of 7 days that there's quite different morphology and progression of the sprouting that's occurring. And if we're quantifying kind of the overlap of the sprouting over the area of the spheroid and how that changes over time, there's significantly, it's not significant here, but we saw that there seems to be more from some of the replicates that we did in this initial pilot experiment. And I think most exciting that we saw is with the kind of larger sprouts that occurred in the fibroblast only spheroid condition, we could do this perfusibility test with dextran and see that some of the dextran does appear to end up in the sprouting, the sprout structures that are where the spheroid is, indicating that this might be something that indicates that it's a useful platform for really thinking more about just spheroid-based angiogenesis here. And then a final thing that we explored with this platform is now wondering, okay, if the fibroblasts are important for inducing angiogenesis and maintaining perfusible network structures, what happens if we put them in a different kind of spatial or spatial organization? Instead of having them in this compacted spheroid aggregate form, what if they were suspended just inside of the PEG hydrogel throughout that entire compartment? So here, this is showing what happens with a perfusibility test when we just had the original spheroid aggregate form of the fibroblasts. And we see that there is decent perfusibility of the networks around the vicinity of the spheroid. Whereas then if we looked at instead suspending the fibroblasts throughout the gel, the PEG gel in the middle, we actually saw this interesting observation that they seem to be pulling on the networks right around the vicinity of that compartment. And then when we did a perfusibility test, the dextran leaked all over into that internal compartment, as well as appeared to be the vessels around the edges seem to be much more permeable as well. And so these are just some of the observations we've made with characterizing the system, but some of the ongoing work that we're currently doing to really adapt it toward endometrial cell culture and ultimately toward our goal of studying endometriosis. One example that I want to show here is we used endometrial epithelial organoids and stromal cells inside the spheroids, as I initially outlined in our in the schematic from before. And this is just re-showing what that adenomyotic lesion looked like with its sort of cystic glandular structure here. And we saw that in some of our experiments that the epithelial cells, which are denoted in the EpCAM, the red stain here, they seem to migrate to the interface of the fibrin and PEG in a way that could potentially be reminiscent of what that structure of the lesions looks like. And looking at the endometrial stromal cell side of the picture, we also had done a preliminary experiment where we created spheroids with the endometrial stromal cells and saw that they, very dissimilarly to the lung fibroblasts, they did not induce sprouting angiogenesis and in fact invaded, breaking down the gel, it seems, and invading into the networks, as well as significantly reducing the perfusibility of vessels in their local vicinity. And so I think these are a lot of observations that we found here and a lot of phenomena that I think we still want to follow up on. But to sum up, we did see that angiogenic sprouts can be observed to extend from perfusible vascular bed into a synthetic PEG-based hydrogel that's tailored for specific cell types of interest. We also saw that we can tune some of our PEG's biophysical properties in order to modulate this extent of angiogenesis. And then we saw that the identity and distribution of the cells that we added into the spheroids or into the spheroid compartment can impact the extent of angiogenesis and local vessel integrity that we see. And so, as I mentioned, there are many things that we want to further explore about the platform itself and thinking about what kind of mechanisms are causing what the differences we see between, for example, the fibroblasts in spheroid form versus in suspension and the role of the macrophages. But as a bigger picture, as we're working toward applying this for endometriosis, some things that we are thinking about adding to the system include how do we support the addition of cycling hormones and other potentially such as pro-inflammatory stimuli? How do we enable the real-time study of immune cells since they're extremely implicated in endometriosis? And then thinking broadly about how we explore how endometrial cells, how they have crosstalk with the vasculature, especially if they're manipulated to mimic pro-inflammatory lesion phenotypes. And with that, there are a lot of people in both of my labs and other supporting labs that I'd like to thank, as well as my funding sources, the NIH NCATS program and the NSF GRFP program. And then finally, there are a lot of people from both of my labs at the conference this week. So these are all of their posters and talks that are coming up for the next couple of days. And with that, thank you for listening, and I would be happy to take any questions.
Hi, that's really very nice work. I have a couple of questions. Firstly, as you know, I'm sure one of the roles of extracellular matrix is binding growth factors and sort of presenting those and establishing gradients. Do you know if your PEG gels will bind and hold onto growth factors?
Do I know if our PEG gels will bind and hold on to growth factors? I am not certain about that part, but I know that one of my next steps for some of these devices was to stain for the extracellular matrix and see what was being produced, at least by the spheroid types of cells that I'm including.
Right. I think that'd be something useful to know. Second question, more observation maybe. When you put the fibroblasts into the PEG gel, it looked like it collapsed the gel. Is that what you were seeing?
Yeah, that's what it seemed to look like for the experiment where I was suspending them within the PEG gel. It didn't seem like that was happening for the spheroid format. And they definitely seemed to, they looked like they were spreading out more when I looked at higher magnification images for the suspended fibroblast condition. So I think that's something I actually only collected those images a few weeks ago. So still trying to think about how to interpret that.
Yeah, it's an issue a lot of us have had using fibroblasts in these devices. They can, under some circumstances, just really become contractile and the whole thing just collapses. So, yeah. Thank you. Very nice.
Thank you.